talk about applications of immune response. So if you look at historical writings, and if you think about to when we started the semester, I talked to you about how the Chinese had this early method to prevent smallpox that was a lot like vaccination. What they actually did was called variolation. They would take the pus from a smallpox patient and dry it, turn it into a powder, and then they would inhale it or place it in the skin. This tended to give individuals a mild form of the disease and gave them immunity. However, this could be fatal, and it made the person who got they were contagious, so they could spread it to other people. This was practiced for quite a while, a few hundred years, and it even became popular enough that it spread to places like Turkey and Europe. However, because it was very expensive and still dangerous, a lot of people didn't stay protected. We also talked about Jenner and how he noticed milkmaids don't get smallpox, they get cowpox. And he actually gave cowpox to some children to see what would happen. And then once they recovered from cowpox, he gave them smallpox. Now, he's famous because they survived and he was able to create the first vaccination. However, we still kind of frown upon giving kids diseases on purpose. Pasteur was later the one who came up with the word vaccination, and it describes a form of protective inoculation. And the only disease we've actually eradicated using vaccines is smallpox. So immunization is the process of inducing immunity, and it's had one of the biggest impacts on human health of any medical procedure. It's an example of how knowledge is power with respect to fighting disease, and it's also had useful applications in terms of immune reactions and creating diagnostic tests. So when you look at how immunization works, immunity can either be acquired naturally or artificially. So it can either happen during normal events like being exposed to an infectious agent or by inducing immunization through an injection. It can also be passive or active. So active immunity comes following an antigen exposure. So it could be natural, again, having an infection, artificial because you had an immunization. When you look at passive immunity, you're actually getting the antibodies from another source. So a natural source would be during pregnancy, a mother actually gives antibodies to the fetus across the placenta. Also, breast milk contains certain antibodies, so if a mother breastfeeds, she gives certain protections to the child. You can also have artificial passive immunity, where you have injections of anti-serum created from other patients. The problem with both forms of passive transfer is that it is not a long-term form of protection. You either need to continue breastfeeding, or you actually need to continue getting injections to remain immune. Now, a vaccine is a preparation of a pathogen or its products. This is used to induce active immunity. So the point is you want to not only protect the individual, but you want to prevent the spread of the disease in the population. Typically, what we're trying to accomplish is called herd immunity. Herd immunity occurs when more than 90% of the population is immune to the disease. This means the infectious agent is unable to spread because there's not enough susceptible hosts. Vaccines are responsible for the drastic decline we've had in childhood diseases in the last 100 years. And effective vaccines should be safe and have few side effects. You want them to give long-lasting protection, but they should ideally be stable, inexpensive, and easy to give to patients. Now, there are a number of vaccines that are given on a regular basis. Others you only give under certain circumstances. And vaccines fall into two categories. You have attenuated and inactivated. So here are some examples of vaccines that are given for viral diseases. So attenuated vaccines actually elicit stronger immune responses, but sometimes cause disease. Inactivated vaccines give weaker immune responses, but they are not capable of causing infections in the patient. So attenuated vaccines actually use a weakened form of the pathogen. The pathogen can actually replicate in the recipient, but the disease is at a low enough level that it is mild or undetectable. The advantages here is that you get long-lasting immunity because the microbes are growing in your body. This can also immunize others because if you spread the pathogen, they also are getting the same immunizing effect. The disadvantage is it can cause disease in immune-compromised individuals, and you can have back mutations like we've talked about in previous chapters, where it reverts to that wild pathogenic form, and it can cause the disease and potentially even kill somebody. So attenuated vaccines are not recommended for pregnant women, and they do require refrigeration to remain viable. Things like the MMR, the chickenpox vaccine, yellow fever, rotavirus are all vaccines that are attenuated. Inactivated vaccines are unable to replicate. So the advantage is they cannot cause disease in the patient. The disadvantage is because the pathogen is not replicating, there is no amplification. Therefore, you have more limited immune responses. This means you end up needing several boosters over the course of your lifetime to get the appropriate level of protection. And oftentimes, these vaccines will contain adjuvant to enhance the immune response. In terms of different types of inactivated vaccines, we have inactivated whole agent vaccines. These actually contain dead microorganisms or inactivated viruses. So they're treated with some form of chemical that kills them, but does not significantly change the epitopes on the surface of either the capsid or the cell. This includes vaccines for things like the flu, rabies, and hepatitis A. You can also have toxoid vaccines. Here, you treat the toxin to destroy the part of it that's actually toxic, and it allows you to retain antigenic epitopes. This is how the diphtheria and tetanus vaccines are formed. With subunit vaccines, they have key protein antigens or fragments of antigens from the pathogen. This allows you to avoid inserting the cell parts that can cause side effects into the patient. The example given of this is the pertussis vaccine. Recombinant vaccines are subunit vaccines that are produced using genetically modified microorganisms. So for the Hep B vaccine, you actually use yeast cells to produce part of the viral protein coat for Hep B, but that way you're only producing the coat and it's being produced by something that's not pathogenic. You have virus-like particle vaccines. These are when empty capsids are produced by genetically engineered organisms. This is how we get the HPV vaccine. You have polysaccharide vaccines, which are made for capsules. Now, unfortunately, polysaccharide vaccines are not effective in children, and they get very poor responses. They work much better in adults, and this is where the pneumococcal vaccine comes from. Conjugate vaccines are where you have polysaccharides. Remember, those are sugars linked to proteins, and what's going to happen is the sugar actually gets converted into T-dependent antigens, so the antibodies can recognize what you're trying to go after. So an example here is the hemophilus vaccine, and this has actually almost completely eliminated hemophilus meningitis in children, as well as the streptococcus pneumoniae vaccine, which is currently under development that might also help with streptococcus pneumoniae. Now, we also have nucleic acid-based vaccines. There's still a lot of research going on with these. Basically, you're trying to take part of the genome, either DNA or RNA, from the infectious agent, just part of it. And what happens is by injecting it into the cell, some of the cells will express the genes for a short period of time. That will induce an immune response so that your cells in the future will target any cells that have those gene sequences. Now, trying to get rid of polio was our next big campaign after smallpox. So when you look at what causes polio, there's actually 3 different types of the polio virus. They all enter via the mouth. And the virus will infect the throat and intestinal tract and then invade the blood. There, it will then invade the nerve cells and cause the disease. So the Salk vaccine was created in the 1950s, and it had inactivated viruses of all 3 forms, and it very quickly lowered the rate of disease, but you needed a series of injections to get maximum protection. With the Sabin vaccine, it was cheaper, it was taken orally, but you still needed 3 doses. Now this produced even better immunity, and you got better herd immunity. The problem is because it's attenuated, it could back mutate, so approximately 1 out of every 2.4 million people who got the vaccine actually had this back mutation happen to get polio. So the Sabin vaccine can cause rare cases of the disease, but it gave better protection. So using this vaccination program, we were really successful at getting rid of polio in the United States and in the Americas. However, we have not eradicated it globally yet, and unfortunately, the US is part of the problem. We almost got rid of it. It was down to just being in like 2 or 3 countries. However, as part of us getting Osama bin Laden, the CIA actually hijacked the polio vaccination program in Pakistan, and when that information was released, it led to retaliation against doctors and nurses in countries that were giving vaccines to poor individuals. Now, some laws have changed in the US, so it's not legal to use medical programs for spycraft anymore, but it is important to realize that it set back eradicating polio by quite a bit. Let's pause here for the next video.